word. Thank you so much, Amanda, for that reminder. So just a couple of quick updates from me, and then I'll call as well if anyone else on the call has any updates that they would like to share. But firstly, ClinGen is in the middle of our annual updates reporting period. I'm sure you are all sick to death of hearing about this on your various expert panel calls, but one hopefully final reminder to please make sure that you're logging into our group and personnel management system, or the GPM, and making sure that your details are up to date, so that you've added in your credentials. This is just from a drop-down list, and also added in your expertise relevant to whichever panel that you're on. Again, this is a drop-down list as we're trying to sort of group different categories of volunteers we have across ClinGen. And then the most important thing, it's all important, but most important thing is please make sure you're updating your conflict of interest survey as well. So this has to be done every year for every expert panel that you're on. So make sure you're logging in and doing that as well. And then the other more fun update, less administrative, more fun, is that next week is the ACMG annual meeting in Salt Lake City. ClinGen, of course, will be there. We're going to have a booth set up in the exhibit hall. We always love to meet our volunteers, so please make sure that you stop past and say hi if you're going to be at the meeting. And also to say that we have a reception on in the evening of March 15. So there'll be some rapid presentations there of various posters and also talks that are on at the meeting, and then an update on our, I want to say it's on dosage, but I feel it's actionability curation.
Actionability, yeah.
So a couple of presentations, but there will also be dessert served in a cash bar there, but please make sure you're RSVPing for that, just so that we can get the catering right. But everyone within the ClinGen community is welcome to attend that reception. If you're on this call, you're part of our community, so if you're at ACMG, please stop by, and we'll put the link for that RSVP in the chat as well. Before we move on to our presenter for today, is there anyone else on the call who has general updates or announcements that's relevant to this forum? I will take that silence as a no. So we can move on to our presentation for today. So we have an external guest presenting today about an external resource, the Gene Curation Coalition database, or GenCC database. Marina is, of course, not external to ClinGen. We all know her very well, but as well as Marina's ClinGen hat, she is also co-chair of the GenCC steering committee. So we'll be presenting an overview and update of GenCC for us today. So I will hand over to Marina.
Great, thanks, Alicia. I will share my screen. Okay, can everyone see my slides okay? I'll assume that's yes. I can't see video, so hopefully.
Yes, perfect.
Great, thanks, Alicia. So as Alicia said, I will talk about the Gene Curation Coalition. Many of you, if you've been in the Biocurator working group for the last couple of years, I usually do this annually, but for those who are new, you may be starting to hear about GenCC in your different GSEPs or somewhere else. So I would like to come and kind of tell everyone about the Gene Curation Coalition, so you have some context there. We also recently published in Genetics and Medicine our marker paper, so feel free to take a look at that. So for an overview, I'm going to give you a refresher on the GenCC for those people who are new, and talk about our Delphi survey where we harmonize terms for clinical validity. I'll then talk about the updates to the GenCC database and tips and tricks to use the GenCC for ClinGen curation. I'll talk about some of the conflict analysis and resolution of conflicts of gene disease relationship assertions in the database, and then some future plans. Feel free to stick questions in the chat too. So it is critical to have curated gene disease databases. I don't have to tell you that, you're ClinGen volunteers, but this really can be seen or demonstrated when you plot the number of genes for hypertrophic cardiomyopathy test from 14 different US clinical labs. This is maybe a couple years old now, but you'll see that the number of genes varies extremely widely, from under 20 genes to over 110 genes. So really it just shows that standards and consensus are needed for which genes are valid disease genes that are ready for clinical testing. ClinGen has been doing this for a long time. ACMG has put out now a statement that says genes with a clinical validity of moderate and above should go on clinical testing panels, and for screening, it should be strong and definitive genes. And this is kind of what I'm illustrating here on the right, that different levels of evidence are needed for different clinical uses. So diagnostic panels, where you have individuals who are affected with disease, might need differing levels or maybe a little bit lower levels of gene disease evidence than predictive tests for healthy individuals who might be undergoing prophylactic treatment based on results. And so we formed the Gene Curation Coalition, and how ClinGen is connected to the Gene Curation Coalition is ClinGen's a member of the Gene Curation Coalition and submits to the database. I'll talk more about that later. But we have a range of public gene level resources and also private gene level resources, all who are members of the GenCC and many of whom submit to our database. And really the goals of the Gene Curation Coalition were to understand the approaches and classification systems of the different curation efforts, to help develop consistent terminology for validity assessment, as well as inheritance, allelic requirement, and mechanism of disease. I'll talk to you today about the Delphi survey we used for terminology for validity assessment for the database. And then we want to post gene curations from any group willing to share with the public. So the GenCC database is kind of like a ClinVar, but for gene curations. ClinVar is for variant assertions. The GenCC database is for gene disease relationships. And really, once all of these were posted, our goal was to kind of mine this a bit and resolve differences and collaborate on gene curation projects. And that's kind of the phase that we're in right now. So in the very beginning, when we formed the GenCC, we really started to compare all of our classification frameworks to kind of understand what was similar, what was different, and do we need a database that helps to look at the differences in gene curations. And really, we showed that even, for example, two different curation groups that use the same system called PanelApp, one's based in England and one's based in Australia, they kind of use the same platform. Even when they compared their gene curations, which are kind of done on the same platform, they found that there were 1,000 discrepancies. They might be major, they might be minor, and they really were able to kind of harmonize a bit, showing that there are some differences and comparisons could be really valuable. And when we did a bake-off and asked a bunch of the original members of the GenCC to perform curations across a range of different genes that are listed here, we found that really the frameworks that we were using for curating gene disease relationships and the names that we were using were very disparate. So we felt that we really needed to start communicating so that we could kind of reach some standards and consensus for the community to really develop this resource and make it comprehensive and understandable so that we were all on the same page. And so when we did these pilot comparisons, we identified reasons for gene curation discordance. Sometimes it was the definition of disease. Some people were just looking at high penetrance versus the inclusion of lower penetrance phenotypes. This could cause a difference in the classification. Maybe the purpose of curation, like I mentioned, PanelApp really kind of asks the question, should a gene be on a particular panel for a particular set of conditions versus ClinGen just curating a gene disease mode of inheritance relationship. Differences in disease or phenotype assignment. Sometimes genes have more than one assertion. You know, you're doing pre-curation, lumping and splitting. Maybe we're classifying on two completely different diseases, and that's why it wasn't consistent. And also not most importantly, but importantly, the date of evidence evaluation. We know that much of the time we're pulling from published literature, and this is why we have recuration guidelines. And some curations are just much older than others. So really understanding those differences and focusing on resolving them was important, and it really required harmonization of both our terms and our definitions, what we were looking at, so we could display them in a database and everyone could understand in the public. And this is just a comparison of our original GenCC members and the terms. And you'll note that there are colors, there are things like confirmed, possible, probable, there are kind of levels of evidence. And even more alarmingly, you'll note that some resources used use colors to mean opposite things. So one of our original submitters used red to mean strong and definitive, and the other used red and panel app like a traffic light system to mean limited and below. So we realized this wasn't a good way to compare things, and this is very confusing for the public. So we really just had to harmonize our terms here. So we performed a modified Delphi survey. And so in the first phase in the development, we worked together to harmonize definitions that did not contain the proposed clinical validity terms. And the second phase, much in a Delphi survey format, we suggested as many terms as possible in the GenCC. And then we surveyed the membership of the GenCC about these terms using a modified Likert scale. So expressing a level of agreement, disagreement, or neutrality for solicited terms. For the second round of surveying, we expanded this to GenCC members and their curation staff. And this really helped to iterate and keep the highest scored terms on the Likert scale. And then eventually we surveyed the international genetics community, and we got 241 responses. And we also included an optional 10-minute explanatory video that kind of just told them many of the things that I just told you. And then in the last phase, we looked at the consensus and also implemented the final terms. So the surveys 2 and 3, survey 2 being the extended GenCC membership and 3 being the international genetics community, actually had pretty high term agreement, 93%, suggesting that the choices of the GenCC are in agreement with the larger genetics community, or at least those that we could reach via survey. So all terms that are used, all terms are used to standardize submissions to the GenCC database. We also added the additional term supportive for less granular submissions. So you'll see that for orphanet submissions as of now. As soon as we have an API connected, OMIM will also start submitting to the database, and they will also be listed as supportive. And to just give you some demographics for the survey, this was the respondent location here, with many of the respondents being in North and South America and the UK and Europe, and a little bit in Australia, New Zealand, and Asia. These were the populations that we surveyed here, with ESHG being the highest number of respondents there. We asked people if they participated in the consortium, and the majority of individuals did not, although ClinGen was the second most answered question, and multiple responses were allowed if you participated in multiple consortia. And then we also asked people about their familiarity with genetics when we were asking them about these terms, in case we had to stratify based on that. And we were happy to see that many people are very familiar with genetics, at least that were answering these questions. Perhaps we should have reached out to those people who are less familiar with genetics to make sure that they also understood the terms, but we didn't have to necessarily stratify by familiarity here. And then we asked people how they would describe themselves with multiple responses allowed, and the majority were researchers, also clinical genetics laboratory directors or staff, medical genetics physicians, and so on. And these are the finalized terms here, and you'll notice they are ClinGen terms because we harmonized to the harmonized GenCC terms. So definitive, strong, moderate, limited, animal model only is a subset of no known disease relationship where there is an animal model, as the public felt that this was an important distinction. No known disease relationship, disputed evidence, and refuted evidence. So the only ones we really had to tweak were no known disease relationship from our original publication, and also adding an animal model only. So next I'm going to talk to you about updates to the GenCC database once we harmonized these standardized terms here and formed the Gene Curation Coalition. So these are the statistics as of today. We have 16,911 submitted classifications on 4,700 or so unique genes from 12 submitters. The database is search.thegencc.org, and here are all the summary classifications visualized, with many of them being definitives and strongs. Also a large orphanet submission of supportive here. And the color scheme across the database is similar. And you'll notice that the ClinGen website also has pretty much a similar color scheme, of course, with the absence of supportive, because we don't have that term. And to just give you some summary statistics, this is from the Genetics in Medicine paper, so this is a little outdated, but we were curious to see the number of submissions per gene. And you'll see that many submissions many genes are just single submissions. There are some genes that have a ton of submissions. Those are actually usually the genes that are associated with skeletal dysplasias, for example, that have a ton of different disease assertions. But I would say that the majority have maybe 5 or less submissions. The average is 2, and the max is 35 here. And I wanted to thank Phil Weller for pulling this today for me. So the use statistics for the last year are shown here, with the map being darker colors being the top users, and these are the top users here. The US, China, Italy, Spain, Germany, France, and the UK. And these users, the number of users per year has definitely bumped up significantly in the last year or so. So now I'm going to show you some screenshots of the database. And if we have any particular questions about the database, I can go there at the end, kind of in a live demo. But this is the gene list landing page here. You'll notice we've got the standardized color scheme that I was talking about here. And it really is kind of a large list of all the genes. You can filter by gene symbol if you want to pull out a particular gene. You could filter by submitted disease. However, at this point, it does have to be exact terms for the disease. We're really hoping to make that disease searching more flexible in the future, and I'll talk about that at the end. You can also filter by a particular submitter if you wanted to see only submissions from a particular submitter. If, for example, you were trying to design a panel, you could filter by the strength of assertions. So, for example, if you only wanted to see definitive ones, you could uncheck all the rest of them, or moderate and below, you wanted to uncheck something like that. It will filter the list for you. And so this top one here is kind of the closed view of a gene submission here, and you'll see like the number of diseases that are submitted for it and the number of submitters and the little graphical representation here. So this one is just one known disease relationship. If you'd like a little bit more information, you can hit the arrows, these gray arrows here, and it will open up and it will show you at least who the submitters are, what their assertion was, and what their disease was that they've submitted on. Just to give you a quick little bite of what you would get if you were to click into that. And so if you want more information, you could click into the details, and that will take you to the gene page. There's also a nice little FAQ help section if you want to learn about GenCC classifications, and this also includes the standardized definitions that we used for the Delphi survey that are all available on the website. So if you click into, if you click on the details button here, you then will get to classify the gene particular page, and there are different tabs that show different views here. So along the top, you can filter anything you want. So if you only wanted to see strongs and definitives within that, if you were on the 35 submissions gene, for example, and you only wanted to see definitives, you could filter on that. You could filter on diseases, modes of inheritance, or submitters. But the tabs here are also different views. So the first tab is by classification. And so you'll see that the stronger classifications are at the top. So the definitive one is here. All the standardized clinical validity terms are here on the left with our colors. We've got the HGNC gene symbol and a link out to that and the ID there. We have the disease. I'll speak more about the disease in a second. We have the mode of inheritance and a little info button in case you're not sure about the abbreviation. The bolded date here is when the gene was evaluated, and the grayed out date is when it was submitted. That's kind of like how ClinVar displays things. And then the submitter. If there's a public report, for example, ClinGen links to the website, to the ClinGen website, you can link to that. You can read about their assertion criteria, which is a document. So in our case, it's the SOP for ClinGen. And you can read more details on the gene page, which I'll show you in a second. So the next tab is by disease. And because we are an international resource and we have the OMIM team and the Orphanet team in the GenCC as members, we have to accept an ontology that includes all of that. And so conveniently, we are harmonized with ClinGen, and we accept we display everything using the monarch disease ontology, but we accept Mondo, OMIM, and ORFA IDs for disease submission. We map them all to the Mondo ontology, but we try to be transparent. So for example, Orphanet, of course, is going to submit using ORFA IDs. So it was mapped to the Mondo term for arterial calcification of infancy, but it was submitted as this Orphanet term. So you'll see if a submitter, for example, TGMI G2P submitted using an OMIM term, and this was mapped to this Mondo term. So we try to be as transparent as possible as to what was actually submitted, even though we do map to disease equivalents, which are the Mondo term, that's the equivalent for whatever was submitted. If you submit using Mondo, we just use your exact Mondo term. And this is kind of the best we can do to harmonize the way that assertions are displayed. It's not perfect, of course, and we'll talk about that in conflict resolution. And then the last one is classification sorted by submitter. So you could filter on this, but for example, in this case, Orphanet has 2, and each one of the other submitters has 1. So kind of self-explanatory there. And if you have a little evidence summary, much like ClinGen evidence summaries, for example, this is displayed underneath the submitter, kind of like it is for ClinVar, and then you would just hit the read more button. If you want to look at an individual submission and get some more information, you can click the more details button at a particular submitter's line. And then you would come to this individual submission page where you've got the gene and the disease and the mode of inheritance when it was evaluated. Some submitters do have evidence and notes, including PubMed IDs, which can be helpful. And then actually there's a separate field entirely for PubMed IDs. This can be very helpful when you're doing ClinGen curations. We'll talk about that. If you want to see how they classify gene-disease relationships, you would click to view their assertion criteria. And there also would be a public link out to their curation page of interest if they are a public resource. Each submitter also has a submitter page on the GenCC website, and it tells you a little bit about that resource that is submitting. It will also visualize all the classifications that they have submitted, their website. And then also, if you ever have any questions about a particular submission from a submitter, there's a public contact, and they know that they're the public contact for each GenCC submitter. And so you could always reach out to them and email them if you have any questions. And then, of course, a link to their assertion criteria. And then all the data is downloadable at this point in different formats. So you can always take a look. And at this point in time, you can do whatever analysis you want now that our marker paper has been published. Eventually, we do hope to have both submitter and user API access if you wanted to, for example, put this in your pipelines in a clinical lab or something. We don't have that right now. We are actually focusing on submitter APIs first, so that, as I mentioned, you could have OMIM on the database. We don't have OMIM right now. I guess in another aside, in the download, we don't have OMIM in the database right now, but in the future, we will not be able to include OMIM in the download because they have their own user license, and that says that here. So they would be excluded from the download. If you're a nonprofit, you just would apply and get a data download from OMIM separately, but we kind of can't go around that. You could easily sign up for email updates too if you were interested in the API. There's a bunch of places on the website that you can hit that button and be notified. And I also wanted to point out that GenCC curations are displayed on the ClinGen website. So if you search for a gene and you click into that gene and you hit the button, so here's of course the gene disease validity curations here, but you hit the button view gene facts, this extra section will open up, which actually has a lot of useful information in it. So definitely look at the gene facts expanded button. But you'll also see a little tiny summary of GenCC classifications. So you'll see in this case, we have 1 limited, 3 strong, 1 disputed, and 4 supportive. Not necessarily enough information for you to really make any choices there, but you could just click the button, read more about GenCC classifications, and it will take you to this entry in the website. We really just wanted to make it possible to show you that the GenCC does have submissions for your gene of interest. And that will lead me to using GenCC for ClinGen gene curations. So you can search the ClinGen website for your gene of interest. We pull in data for genes that haven't yet been curated by ClinGen. So if you have been assigned a gene, it hasn't been curated by any other GSEP, you can search it you can open up these gene facts and see if it's been submitted to the GenCC database, or you could go to the GenCC database directly, your preference. Then you can link out to the database, and if the disease appears correct or appears to be the disease that you would be evaluating, you can click on individual submissions to use PubMed IDs for curations. Much like we say in the SOP, I often pretty much exclusively encourage using primary data for your curations. You would never want to say so-and-so said that this was definitive, therefore it's definitive. You always want to look at their references that they're citing if they have them. Some groups do use stronger definitive assertions in the GenCC database to justify scoring functional evidence. This happens in the syndromic GSEP that I work with. So for example, protein interaction for interacting partners. A lot of times you know that something is maybe strongly or definitively associated with disease, but you haven't curated that gene yet. You can look in the GenCC database and see if multiple submitters say that something is strong or definitive for the same disease and potentially score that as functional evidence. But please check with your GSEP ahead of time. The comfort levels of GSEPs definitely differ. So now I'm going to talk to you a little bit about conflict analysis. So we've set up this database. It has a bunch of different assertions, 16,000 or so assertions on gene disease relationships. But how are we doing as far as, are we saying the same things? Are we comparing apples to apples? So I'm going to tell you a little bit about our conflict analysis and discrepancy resolution we've been doing now that we have this database set up. So these are the curation conflicts that we define here. So supportive is not included, which is why it's kind of above, but anything that's definitive, strong, or moderate versus anything that's limited, disputed, refuted, no known disease relationship, animal model only. That's what we define as a conflict. However, it has to be the same, you have to be talking about the same thing. You have to be talking about the same disease and the same mode of inheritance. The same disease is kind of nebulous, right? Because some people curate to a higher level disease term, some people curate to the exact mondo disease term. So that does require some manual curation and discussion, but we are trying our best to work on identifying real conflicts. So in order to do this, we start with a computer-generated report, and then I review the report because we know that disease synonyms kind of have to be manually reviewed. So we have different levels of automatically calculated conflicts. So the first one is what we call a level 1 conflict, where the gene matches and we ignore the disease term and the mode of inheritance. So we're basically just saying, here's an example here, two submitters, the gene's the same, we're ignoring the disease, which in these cases are not the same at all. We're ignoring the mode of inheritance, which in this case is also not the same, but we're saying this is a limited assertion and this is a moderate assertion. Therefore, it could be a conflict. Please, someone look at it. And this is really our least granular way to try and identify as many conflicts as possible. So you'll notice this is from the Genetics in Medicine paper, so I don't have a completely updated statistics, but from that time, I think it was maybe 12,000 or 14,000 submissions in the database. We had 610 genes, so 13.4% of genes in the database had one of these kind of really not very granular conflicts. Most of these wouldn't turn out to be real conflicts, but we want to try to cast as broad of a net as possible because I can't review all 16,000 submissions in the database. So then we went to something a little bit more granular, and we call these level 2 conflicts. And so these we matched on gene and mode of inheritance and ignored the disease term, which is maybe a bit more realistic because people might not exactly always be using the exact same disease term. So here's an example here for submitter 1 and submitter 2. The gene's the same. The disease may be a synonym here. The mode of inheritance is the same. And this person's saying strong and this person's saying limited. So this might be more realistically an actual conflict and is a bit more granular than the ones above. So in this case, and of course, these would all kind of fall into level 1s. It's just a more granular filtration here. So you would expect fewer conflicts in this bucket. So we have 488 genes in this level 2 conflicts bucket, which is about 10.7% of the database. And the most granular ones, which are the easiest ones to computationally identify, are level 3 conflicts where everything matches. The gene, the exact disease term, or at least the equivalent, and the mode of inheritance, they all match. So you'll notice here, same gene, same exact disease, same mode of inheritance, some person saying strong and some person saying limited. So this is the most granular, and you're going to miss those submitters who use disease synonyms when you look at level 3 conflicts. And so as you would expect, these are the smallest number of conflicts, so 246 genes, 5.4%, but these are bona fide, these are real conflicts. So I'm guessing with these type of buckets that the number of conflicts kind of does fall somewhere in the middle if I were to review all of them simultaneously and count real conflicts. But this is an overestimate and this is an underestimate, of course. So for conflict resolution workflow, we have that automated conflict report with all the levels that I just talked about, and then a manual curator review. Most of the time it's me or one of the Broad ClinGen biocurators. And then we make conflict reports for different submitters. And most of the time, the conflict reports will include simple conflicts, which means your submission is out of date compared to other submitters, or your submission is an outlier submission. And most of the time, this would mean there's 3, for example, there's 3 total submissions. You say limited, the other 2 people say strong. Can you please look at your outlier submission? And this generates a submitter-specific conflict report for offline review, and then the submitter updates the GenCC database. If there's a more complex conflict where all of the submissions are around the same date and maybe it's split, 2 people say limited, 2 people say strong, these are things that need to be discussed during the GenCC meeting, and then we consensus vote. And we don't want to tell people specifically, like, you need to submit this as definitive, because we feel that's not appropriate. We are really talking about judgments on the conflict line. So we'll make judgments of limited or below, or moderate or above, kind of to resolve those conflicts. And it's kind of up to the submitter to go back and evaluate the data and decide what they're most comfortable with. And the submitter, of course, has an active part in this discussion. So we're not just telling people you have to change your submissions. We're asking them to vote. We're asking them to say why they classify things the way that they did. So for conflict discussion of these complex conflicts, I present an overview of the gene and all the combined evidence for the curation that I can find. I try to notify submitters ahead of time if possible so that they can prep and comment on their curation, or if not, I at least try to pull what they've included as their evidence from the database. And then the combined members of the GenCC decide on an appropriate evidence level that's less granular, like moderate and above or limited and below, and offer guidance to the submitters. And then submitters use this guidance to reevaluate. So this is an example of a complex conflict that we discussed on a call with CAM-K2G, where we had 3 limiteds, the pinks here, 1 strong and 1 definitive. So it's like, what is the truth here? And if you were from the genetics public kind of trying to use this assertion, what's the truth? So we presented all of this and discussed it, and our final decision actually was limited or below. So we suggested that all those submitters that had submitted higher than that go back and reevaluate and resubmit. And we also suggested that lumping and splitting needed to be reevaluated by submitters. And as of now, we have resolved 21 complex conflicts. And this is kind of the summary of those resolutions. So 9 of them ended up being limited or below. 2 of them actually ended up being borderline limited or moderate, which I feel like as curators, you understand there is kind of that gray area and the points in between. For example, if limited in the ClinGen system is 1 to 6 and moderate is 7 to 11, what happens if you end up with a 6.5? Some people are going to think it should be limited. Some people are going to think it should be moderate. So we did have 2 that we couldn't decide whether things should be limited or below or moderate and above. And so we allowed those conflicts to still exist, with some people saying limited, for example, and some people saying moderate. If you had a definitive submission, that maybe was not so okay in these cases. And then 10 of the conflicts ended up being voted as moderate or above. So really kind of a spread there. And then I'm going to end with some future database enhancements. So as I mentioned in API, so we need this to ingest the OMIM data, and this date should be changed. It should be later in 2023. And then much like ClinGen and the ClinGen website, we're hoping to have a login screen and dashboard for saving searches, following genes, et cetera. because Phil also develops and maintains the ClinGen website, you'll notice that the look and feel is similar, and there might be shared features across. And then disease searching flexibility. I told you right now you have to use exact terms, or only exact terms that are in a particular disease name will come up when you search. So we're hoping to add some flexibility to that, because as I showed you for conflict and analysis, people are using synonyms with their submissions. So we're hoping that Mondo hierarchies will be incorporated into the database to really allow users to search by any term in the Mondo tree, or synonyms, or HPO terms. And this is also being discussed for the ClinGen website, but we're hoping, because it's a bit of a smaller environment, that we could potentially deploy it in the GenCC database first. And also potentially a more granular capture of mechanism of disease. This has been a request from the community to the general GenCC email address for quite some time. But as you all know, it's quite difficult to decide what to call mechanisms of a disease, or to even develop curation frameworks for that, which we've discussed quite a bit in ClinGen and this kind of ongoing work. And we might consider even a submitter that points out candidate genes in ClinVar with likely path or path variants that are not yet in GenCC or OMIM, that really might show you if there are some limited or supportive candidate genes out there. And with that, I want to acknowledge all those members of the GenCC steering committee and all those members of the GenCC, and thank them for all their submissions to the database. And then Scott and Phil for their work on the website and the database. And with that, I will stop and potentially take questions, or I can show you, click around in the database if that would be helpful.
Are there any questions for Marina?
So I see one. Is it indicated on the interface whether a gene with conflicting assertions has undergone conflict resolution, and what the determination is? That's a great question, Carla. We have discussed potentially including a tag on the database to show that something is either undergoing conflict resolution or has undergone conflict resolution. It hasn't been deployed in the database yet, but I'm going to say yes, eventually we will have things tagged, just not as of yet.
So any other questions? Otherwise, your suggestion then, Marina, of like going into the, maybe just quick, I'm not sure how familiar people on the call are with the interface. So maybe just this is how you navigate to the things that you're going to be interested in, just quickly. And then, yeah, if there's no other questions, we can give people some time back to their day.
So there are two ways to get to the database. There's first just the website, and this is just the GenCC website here, and it will tell you a little bit about the marker paper. We had a podcast that for genetics and medicine that Heidi and I were on. It will tell you about some of the projects that we've done. It will tell you a little bit about the steering committee, the goals of the GenCC. It'll tell you a little bit about the members. And this will talk about, for example, we've partnered with HGNC that they're going to prioritize those genes in the database that are strong or definitive, stabilizing the gene symbols for those genes. So that just a little bits of news and articles about that. And then if you want to see the database from here, you just click GenCC database. However, you could also just go directly to it with search.thegencc.org. And this is the database, as I mentioned here, and it's kind of like just a giant line of all the genes. It only displays so many results at a time, so it's a bunch of pages. But you can filter by gene symbol, and it will pull up. You could filter by the submitted disease. So for example, when I was writing my R01 and I wanted to find different neuropathies, you could type particular diseases here, but it has to be exactly within the disease name, as I mentioned, and it will pull up some genes for you to help with gene list creation, for example. You can filter by submitter if you only want to see a certain submitter, or if you don't want to see a particular submitter. I will add, though, that these filters, when you click them, you may not think that very many things have changed, but that's just because this is a combined view. So for example, if you didn't want to see anything from ClinGen, these genes may still have a ClinGen submitter, but because they have other submitters, they're still shown here. That if I filtered out ClinGen, it's really only the submissions that are only ClinGen that disappear, if that makes sense, just because these are combined lines here with multiple submitters. Kind of the same thing goes for filtering by clinical validity here. So if for some wild reason I just wanted to discover genes, I didn't want to see those that are definitive, you'll notice that there are still some genes with definitive submissions here, but that's because they also have other assertions. So we didn't want to make those disappear, but those that were definitive only, for example, have now gone away, if that makes sense. There is help documentation with common questions. What's the GenCC? What are the goals? How do I acknowledge the GenCC for database use? And then some people have asked how you can join the GenCC. So we have some very specific terms if a lab or a submitter wants to submit to the GenCC. And then if you want to learn about the classifications, we've got a little bit of info about the Delphi survey and then all of the definitions here that the harmonized definitions we've drafted. And then if you want to go into a particular gene, go into one that has a bunch of information. So you just click details and these are all the views. So here is maybe where filtering is a little bit more helpful. If you're like, I don't want to see limited genes. That will make the limited ones go away, although it will tell you it's been filtered. It will also kind of tell you up there. Or if I'm only interested in one of these diseases, I'll get rid of it. Or if there were multiple inheritance patterns, something like that. So the first one's by classification, and then the next one is by disease here, and then by submitter, if you want to see that there are multiple submissions. If you go to more details, you'll notice that the ClinGen evidence summaries aren't here, but that's because we link to the public report, which will take you exactly to here. And one note about ClinGen submissions in the GenCC, we aren't connected by API right now. And so Phil will just do giant data dumps of the entire database every so often. So you will notice slight discrepancies between what's been submitted in the ClinGen data set for GenCC versus what's on the ClinGen website. So I do suggest if you're looking for a ClinGen curation that you should still go to the ClinGen website because we don't have an API that if someone publishes to the website, it's updated on GenCC in real time. That hopefully should be deployed soon. We'll be one of the first groups to test the API just because the same developer works on both websites. But as of right now, there is a fork in the data set. And then if you wanted to go to submitters, you can learn more about the submitters, their assertion criteria, if you want to filter their submissions, and who you should contact. And then if you want some statistics, this is the screenshot that I showed you before, and also the statistics across the different submitters. If you want to have data downloads, just click the download button and here they are. If you want to contact us, we're GenCC@thegencc.org, but I check that email address, so just contact me. And about just takes you back to the page here. And so that's kind of a brief overview of the GenCC website.
Excellent. Well, if that introduction to the website itself hasn't raised any questions, we probably all have 15 minutes back to our day. Just wait and double check that no one had questions. But yeah, thank you so much, Marina, for that overview of what is a really valuable resource as it is already. And then it looks like there's lots of exciting things planned for the future, which is just going to make it even better. So thank you so much.